Okay, so we shall be doing a revision in biochemistry. Okay, so first of all, we are going to start with physiological buffers. So the first thing that we're going to start with, physiological buffers. So what is a buffer? So by definition, a buffer is any substance that resists change. Okay, so any substance that resists any change in pH. Now, there are different buffers that are so important under biochemistry that we must remember. The first buffer to remember is the bicarbonate buffer. Okay, bicarbonate. So this is the first buffer to remember. The second buffer is called the phosphate buffer. Very important. Phosphate buffer. The third buffer to remember is simply plasma proteins. Plasma proteins are also potential buffers. Okay. So remember that they resist any change in pH. So they are very, very important. Okay, but which one amongst these is the most important buffer? Most important buffer is the phosphate, the bicarbonate buffer. Okay, the bicarbonate buffer. Which one is the most important buffer? Bicarbonate buffer. The phosphate buffer is important for maintaining intracellular intracellular pH. So which buffer system is important for maintaining intracellular pH? The phosphate buffer. Which one is the most important buffer? The bicarbonate buffer. All right. So now we look at the ranges of the pH, the partial pressure, as well as the bicarbonate range. So the normal range for the pH, the pH is supposed to be 7.35 to, let's say, 7.45. This is supposed to be the pH. The partial pressure for carbon dioxide is supposed to be 4.5 to 6.1 kilopascal. This is the normal partial pressure. How about the bicarbonate concentration? Bicarbonate concentration is supposed to be 22 to 26 millimole per liter. Okay, so we now look at so anything that is above that is said to be uh, abnormal. Now we look at what we call metabolic acidosis. What is metabolic acidosis? Metabolic acidosis. When does it occur? So, metabolic acidosis is a decrease in the pH. So, this is just decrease in the pH. Okay. What causes it? It is caused by decrease in bicarbonate concentration. Okay. Caused by decrease in bicarbonate concentration. So bicarbonate is supposed to neutralize the acid. So if you have few bicarbonate, means you are going to have a higher concentration of you are going to have a rise in H plus ions. So it means that the pH is going to be lowered down. Okay, let's look at the symptoms. What are the symptoms of metabolic acidosis? So symptoms, number 1, nausea, vomiting, and fast breathing. Fast breathing. Okay, so these are the symptoms for metabolic acidosis. Okay, we look at the diseases which are caused due to poor acid-base balance. Okay, the diseases which are caused due to poor acid-base balance, we have diarrhea. Okay, so we have diarrhea, we have diabetes ketoacidosis, diabetes ketoacidosis. What else? We have lactic acid. Okay, we have lactic acid. Now, so these are the diseases which are caused due to poor acid or base balance. Okay, let's look at metabolic alkalosis. Metabolic alkalosis. Okay, metabolic alkalosis. Okay, what is it? Decrease. Sorry, this one is opposite. So it will be increase in pH. What is causing the increase in pH? Remember, it will be due to increase in bicarbonate. So if bicarbonate level increases, it means the pH level is going to increase. Hydrogen plus is going to decrease. That's the concept behind. Okay, so it occurs mainly due to loss in H plus ions. Cause, look at the causes. Number 1, we have vomiting. Vomiting is number 1. Number 2 is nasogastric sanction. Nasogastric sanction. Nasogastric sanction. Okay, we look at another one, which we're calling as respiratory acidosis. So what is the cause? The cause is increase in partial pressure for carbon dioxide, which is hypercapnia, which is also known as hypercapnia. Okay, so increase in the partial pressure for carbon dioxide means a decrease in the partial pressure of oxygen, which is leading to hypoxia. Okay, hypoxia. Now we look at principles of enzymology. We look at the principles of enzymology. Okay, so this is also important. Principles of enzymology. Okay, so what are enzymes? Of course, we know that enzymes are just definition. Okay, molecules that speed up the rate of chemical reaction. So those are enzymes. Okay, they speed up the rate of chemical reaction. Okay, so now we look at the internal classification of enzymes. We look at internal classification of enzymes. Internal classification of enzymes. Internal classification of enzymes. So now the internal classification of enzymes. So under this topic, it's very important to remember that we have different enzymes. So we are first of all going to talk about the oxidoreductases. Oxidoreductases. What do they do? Okay, so from the word oxidoreductases mean they catalyze oxidations and reductions. Okay, so catalyze oxidation and reduction reactions. They catalyze oxidation and reduction reactions. Transferases. From the word transfer, what does this mean? Transferases These, they just characterize the transfer of a group, okay, let's just say other than hydrogen, between a pair of substances. So between a pair of substances, they characterize the transfer, okay, they characterize the transfer of groups between a pair of substances, with hydrogen being an exception. We look at the liases. Liases, what do they do? Well, these are just enzymes that they characterize cleavage. Don't forget, these they characterize cleavage of carbon to carbon bond, carbon to nitrogen bond, carbon to oxygen bond. Okay, these are the reactions that they characterize. We look at the hydrolysis. Hydrolysis, what do they do? So from the term hydrolysis, they characterize hydrolysis. They characterize hydrolysis of esters, ethers, okay, peptides, etc. It's very simple to remember. We look at the isomerases. There are 6 isomerases. Isomerases, what do they do? So these, they characterize geometric, structural, they characterize geometric structural change within a molecule. So they characterize geometric structural change within a molecule. Okay. Now we look at the ligases. So this should be number 6. What do they do? So they characterize joining together of 2 molecules. They characterize joining together of 2 molecules. So the best way to remember this, remember that from the same DNA, the ligases are the ones which characterize the joining of 2 molecules, joining of 2, let's say phosphodiester bonds. It's these same ligases. Isomerases, they just characterize the geometric or structural change of a particular molecule. Okay, so having done this, let's now look at the factors that affect the rate of reaction. Factors affecting the rate of reaction, we have number 1, temperature. Number 2, we have reaction concentration. C, we have the pH and the presence of an inhibitor. Presence of inhibitor. Okay. We look at the enzymes important for regulating blood glucose after meal. Okay. Enzymes important for regulating blood glucose after a meal. What are those enzymes? They're simple. The first one is the hexokinase. The second one is the glucokinase. So hexokinase, from hexos, glucokinase, glucokinase. Very important to remember this part. Okay, what are some of the examples of covalent modification? Examples of covalent modification. Examples of covalent modification were, number 1, we have allosteric modification. Allosteric modification. Number 2, we have effector molecule. Okay. Sorry. Okay, so the first one, examples of covalent modification, number 1 that we have is the allosteric mechanism. The second one is called covalent modification. The third one is phosphorylation of group glycogen. Okay, I think we have 2, but there is a point which I want us to take note. The first point is that phosphorylation of glycogen, phosphorylase, increase its activity. Phosphorylation of of phosphorylation of glycogen, synthase, reduces its activity. Okay, very important to take note. All right, so we can now look at what we call signal transduction pathway. Signal transduction pathway. So, what is to transduce? Transduce is just to convert or change into different energy. To convert or change something to different energy. So signal transduction is the transmission of molecular signals from a cell's exterior to its interior. Now let's look at the stages. What are the stages that are involved? Okay, so the first stage is called the reception stage. There are three: reception, second one, transduction, the third one is the response. Reception, transduction, and response. So how does it go? Okay, so we're going to break it down this way. The first stage is the reception stage. Okay. Reception stage. See the reception stage. So now what happens during the reception stage? During the reception stage, we have the binding, of epinephrine to G protein linked receptor. The binding of epinephrine to G protein linked receptor. Okay, that's the first stage. The second stage, immediately this happens, immediately epinephrine binds to G protein linked receptor, we enter the transduction stage. We enter this transduction stage. Okay. Transduction stage. In the transduction stage, inactive G protein, okay, this inactive G protein is going to be converted to active G protein. Okay, then inactive adenyl cyclase is going to be converted to active adenyl cyclase. Then ATP is going to be converted to cyclic AMP. Now we reach what I like calling the IPK. We now reach the IPK. So in the IPK, let me just write it here. Okay, so we now reach the IPK. I'll be using this. So we're going to have inactive protein kinase. After ATP is converted to cyclic AMP, we have inactive protein kinase, inactive protein kinase is going to be converted to its active form, which is the active protein kinase. Okay, active protein kinase. Like that. Then from there, remember it's the IPK, inactive phosphorylase kinase, inactive phosphorylase kinase is going to be converted to its active form, which is the active phosphorylase kinase. Then we are going to have the IGP. So inactive glucose phosphorylase. Okay, so we're going to have inactive. We now enter the IGP. IGP comes after active phosphorylase kinase. So inactive glycogen phosphorylase is going to be converted to its active form, active glycogen phosphorylase. Then this active is the one which now is going to give rise to group which is going to give rise to glycogen, and glycogen will give glucose 1-phosphate. Okay, glucose 1-phosphate. So this is how it goes about. It's very simple.